Good evening, everyone, and welcome to the NF2 Accelerators Meetup meeting on December 5, 2022. Tonight, I will give you an update on the NF2 research portfolio. My name is Salvatore La Rosa, and I'm the Chief Scientific Officer at the Children's Tumor Foundation. So our approach for not only the NF2 portfolio, but also in general for the neurofibromatosis and schwannomatosis portfolio has been a mix of different components, different type of, let's say, technology. We've been using gene therapy, we've been looking at gene therapy, and we also been looking at drug treatments. But together with that mixed approach of different technology that is not limited to gene therapy and drug treatments, I just want to highlight two of them. We also been looking at what are those tools that we need to use to accelerate discovery to clinical benefit, and what are those new discoveries that are leading to better treatments. So in the realm of basic research. And last but not least, opportunities with pharma and industry and developing new business model where the foundation can actually unlock those assets that are in the pharma and biotech sector, where we can unlock to the benefit of the patients. We are just in the middle of completing our strategic plan and setting our goals. And as a reminder, this is just a summary of what I already said. We're trying to invest in new discoveries, from brilliant ideas to investable projects. We're trying to explore all treatment options in a portfolio approach, but also what is very important, we are trying to progress what is promising research. I'll show you tonight that there are a few examples where we funded very good science that kind of advanced and proved to be very, very effective. And we are not stopping it. We are trying to move it forward with other grants to progress. And then together with, let's say, the portfolio, which is in blue, we're also trying to build that infrastructure, which I said, you know, where is to accelerate the path to the clinic. One is, for instance, to invest in advanced promising research that only that has a clear path to the clinic. Sometimes industry or companies give you like, oh, we can test this compound, but that compound is on a dead end. We don't want to do research twice. So we're trying to focus now on everything that has a clear path to the clinic. And also increase the capacity for more clinical trials. You'll see platform trials. We'll talk more and more about those, but also patient recruitment, patient education, and clinical care, because it's important that we have excellent clinical care if we want clinical trials. And as I said before, unlocking corporate opportunities, maybe by using venture philanthropies. And you'll hear more as soon as those strategic plan and goal setting will be finalized early next year. And here are, you know, the ideal, let's say, workflow of how the NF2 accelerator, in this case, is looking at a list of different approach of ideas, drug therapies, gene therapy, creating animal models that are available, and creating corporate investments that funnel those ideas that will generate assets or drugs or research outcomes that will use the clinical trials, as I said, innovative basket or platform trials like the Intuit trials that you know already, the EU Pearl, that is an initiative in Europe to create platform trials for all forms of NF, but also leveraging our registry, the clinic network, that will create that infrastructure where all of these results coming from the NF2 accelerators idea will plug into, and hopefully will get to the finish line with an approved medicine for NF2. As you see underneath, I also highlighted some of the drugs that are now in the pipeline that CTF was able to bring to the clinic due to investments that we made. And the brigatinib was the first one. Neratinib, I'm going to talk about that, is the second drug that was just approved in Intuit, but also other drugs that I hope they're not yet there, but they're on the path to become, clinical trials. So the menu of today is I will talk about newly activated NF2 grants, specifically these three from Hardin, Amun, and Castellanos. Then I will give you an update on a new grant to Leizu on Losartan. We heard from Dr. Leizu earlier this year about the Losartan research that she actually came up with. And she said that, there were important steps forward. So tell you more about that. We had a very, very interesting proposal that we just approved for Dr. Morrison and Dr. Jean, collaborative grant on NF2 gene replacement. This is something completely new. I will give you some updates on the NF2 Therapeutics, the gene replacement study that we are conducting with this company that is 100% focused on NF2. I'll provide you with some updates on the Intuit trial for NF2, and then we'll look also briefly, not in depth, other ongoing NF2 research, just for you to know that there are a lot of things moving. Okay, so let's go to the first new grant. And this grant was, it's a Young Investigator Award that we awarded to Haley Hardin. She is a graduate student and working to get her PhD at the University of Central Florida under the supervision of Cristina Fernandez Valle. And as you can see here from the objective of her study, she's looking at the potential for NF2 patients to become resistant to single agent kinase inhibitors, which is very high because patients are exposed to one single drug in a chronic manner. And this means that when you take one drug for a very long period of time, sometimes your organism, your body get used to it and doesn't respond as the first time you had that drug. So this is a risk that it's very palpable everywhere for any type of treatment or drug exposure. They want to looking at specific combinations. In this case, is a BRD inhibitor with a MEK inhibitor. So the MEK inhibitor is exactly the same MEK inhibitor used in NF1, which is, I think, selumetinib, and the other drug instead is in combination. The study will investigate the drug resistance. So when you're exposed to either a single agent, either the BRD4 or the MEK inhibitor, the cells don't respond on a long period of time, but the combination should ameliorate the response rate, also allowing for those reductions. And those two mechanisms are approved already from very preliminary results to be very effective in NF2. So she will look at this. She's just started this July, and of course the award is for 2 years. And because she's a graduate student, the award is $64,000, meaning $32,000 a year. Another very, very interesting and more close to the clinic award. In fact, this is a drug discovery initiative that we approved for Dr. Sylvia Amun at University of Plymouth in UK. And she's testing 2 compounds. One is an AXL inhibitor, and this is the code BGB324. And the other one is a MERTK inhibitor that are 2 compounds. And she's testing this in animal models. So it's a very straightforward experiment where these compounds will be tested in this, and the goal is to examine the efficacy of these inhibitors in mouse model of NF2 schwannoma, with the goal that if there is any positive outcome, this drugs or the proof of concept will be validated so that we can enable testing of these inhibitors in human clinical trials. So this is a 1-year award, again started in June this year for $85,000, and this group has been very prolific in generating new ideas and testing drugs. So their animal model and their facilities is very, very effective. And here we go with one of those examples that I was mentioning before, when I said that if there is a very positive outcome in one of the studies that we have funded, we're trying to make that study continue progressing, so not stopping. So in here, I'm referring to Elizabeth Castellano, Dr. Elizabeth Castellano from Barcelona, Spain. And in 2020, 2021, CTF awarded her with a grant for the evaluation of and the use of antisense oligonucleotides as personalized therapy for NF2 patients. Well, that study in particular, which was a cell study, produced very positive results. So they were able to create an antisense oligonucleotide for NF2. Now they're progressing their research, and we awarded them just this year, a few weeks ago actually, a new award where they are developing NF2 iPSC cells model to test these oligonucleotides, antisense oligonucleotides, as a personalized therapy for NF2 patients. This means that they created the antisense, and now they're also creating the test model, so the cell lines where this can be tested. And having these cell lines ready as a non-exhaustible source of cell lines will allow them to move forward with the validation of this therapy. So here is a full description of their award, which is again a drug discovery initiative. As you can see here, they propose to generate these cells with NF2 altered at exon 11 and test the efficacy of these therapeutic molecules that they already developed in their previous award. So it's the next step, and we hope that in a year we'll be able to validate this into these newly developed models. The other very good thing about this award is that these models, these cell lines, will be useful for also the other type of testing, not just this antisense, but will be useful as a source of cells for the entire community. Here we go again, and with Dr. Lei Zhu at MGH and Harvard Medical School. If you remember from our previous talks and updates, she completed very successfully this award that CTF gave her in 2020 for $85,000, where she was looking at reprogramming the tumor microenvironment to enhance anti-tumor immunity, and in NF2 vestibular schwannomas treatments. This actually led to the discovery of Losartan as a treatment that can be very useful in hair restoration or hair preservation. So now that research is progressing now with a new award, which was approved a month ago, where she will profile Losartan, induce changes in tumor microenvironment and inflammation in NF2 vestibular schwannoma patient sample. So from, let's say, a DDI, from a drug discovery initiative study, we're now moving into clinical study. And this study for $150,000 will be completed in 2023, 2025. She will look at samples, so patients samples that were exposed to some sort of Losartan, so antifibrotic type of drugs, and she will profile that in those samples. In preparation and building that type of data, just yesterday, I was attending a meeting from the NF Clinical Trial Consortium, and Scott Plotkin presented a letter of intent. So the study was presented for a clinical trial concept of Losartan in NF2. This letter of intent, or this way of presenting these clinical trials, is very slow. I mean, it takes a few months to get on board, because the first protocol, the first proposal is presented to the consortium, and then via few refinements, then the final trial is either approved or modified. But it's very, very important that the first step towards a clinical trial of this agent is already approaching the consortium for a start. And here we look at the specific of the award that we gave to Dr. Zhu, where you can see here the study provides a rationale and critical data for the clinical evaluation of the effect of Losartan on hearing function in patients with vestibular schwannoma. And as I mentioned before, she also wrote that Dr. Scott Plotkin is planning a clinical trials, which was presented yesterday at the Clinical Trial Consortium. And now they propose in preparation for this trial, which I already told you it will take a few months, to start confirming the findings in mice with NF2 patient samples, to also identify biomarkers that may improve the ability to predict hearers at risk for hearing loss. So very, very exciting that we started with very early research and proof of concept. Now we're moving into clinical trials, and that clinical trials already is planning for full clinical trials by the DoD and the Clinical Trial Consortium. So this concept, it's moving forward. Here we go. So another piece of research that is moving forward, that is making progress. This time, the opportunity comes from NF1. As I said before in my topic content, we have an opportunity for gene therapy in NF2 by building on a very good and very solid results that we obtained in NF1. I'm not sure you know about Dr. Samantha Jean from University of Sydney. So CTF awarded her a grant for $240,000. This was one of the first grants we awarded in gene therapy for NF1, where she was developing a new proprietary adeno-associated virus variants. So she's developing this virus, and this virus are loaded with a payload, and the payload is the gene that needs to be fixed. So she developed the container, so let's say the vehicle, which is this virus, and she actually had very good results. She was able to obtain a very efficient virus that can actually transport some of the genetic materials to the target cells, which is human Schwann cells. That is completed. So the study was completed. But now we are also progressing research, because we awarded at the beginning of 2022, first of all, try not to stop this type of research. We awarded another award for very expensive, $196,000, if you think, because we wanted to test these novel bioengineered vectors that are targeting Schwann cells in a non-human primate model. This is very, very important, because the proof of concept that is in cells needs to be actually validated, especially for gene therapy, in non-human primates, and then we'll have a proof of concept that vector, that virus really works and really delivers that payload. In this case, NF1 to the Schwann cells. So you will ask me, where is the opportunity here for NF2? Well, here is the opportunity. If you think about NF1, NF2, and schwannomatosis, the common element that are between the three syndromes is that the cell type, so the cells where tumors, in case of NF1, we're talking about neurofibromatosis. In the case of NF2, we are talking about schwannoma, and also for schwannomatosis, we're talking about schwannoma. All of these tumors arise from Schwann cells. So the opportunity here is that once we have a vector that targets Schwann cells, then the question now becomes, what is the payload? And the payload can be NF2, can be NF1. So here is the proposal. So why don't we try to build on this excellent vector variant of the virus and charge it with the NF2 genes? So the idea was that, we actively connected Dr. Jean with Dr. Helen Morrison in Germany, and she is a very, very expert NF2 biologist that has already proved in vitro, so in cells, that if we restore the NF2 genes, the cell basically function normally. So we basically, let's say, solved many of the problems of NF2 tumors. So this was a new proposal that we just approved. So here we see, and it's a very big proposal. It's over $1 million in 3 years. And why is that expensive? Is because we are doing basically what you saw for NF1, we are trying to do it also for NF2. So they are developing an adeno-associated virus-based genome editing approach for the treatment of NF2. So this proposal will start and will build from the base vector. So we'll take advantage of newly developed capsids with superior delivery to primate human Schwann cells that we saw for NF1, and trying to load them with a CRISPR-based gene editing that will attempt to restore physiological NF2 gene function within cells to permanently cure the cause for NF2 schwannomas. So we approved this grant in 2022, and it's going on. It will last for at least 3 years, but we hope that those results will show us the way forward also for NF2 in using this carrier. And we switch gear, and well, not switch gear, but we'll stay in the gene therapy space. And what I want to give you an update is on NF2 Therapeutics. You probably heard, and or if you don't, Michael Wooden is the CEO of NF2 Therapeutics. He has updated this group and in general other groups on what they're doing for gene therapy, and we have a grant with them for about $400,000 that we started years ago. But what NF2 Therapeutics is working on two different things. So we are collaborating with CTF on developing a disease-modifying gene replacement therapy for NF2, and also the company is working on an antibody drug conjugate, which are called ADC, for meningiomas, which are the leading cause of mortality anyway in NF2. So the current objective of NF2 Therapeutics is to de-risk the gene therapy and ADC programs, and by de-risking meaning having a proof of concept to then scale up and build a company around it, even bigger than the current company, because of course they will need like serious investments from investors. So the NF2, the key achievements for the gene therapy, so the adeno-associated virus NF2 program are quite substantial. I mean, first of all, the program has been put on hold and delayed for many different reasons. One for all, you know, this was awarded in 2019 and COVID hit. So that was a huge bump on the road, but also there were some problems with some of the studies that were done that needed to be redone because they were not accurate. So finally, after, I would say, 3.5 years, we are at a very good point, I would say. So first of all, they already are at the second generation of AAV gene therapy constructs that were scaled with commercial supplier capable of large-scale, good laboratory practice or good manufacturing practice, meaning this scale can actually sustain clinical trial. So meaning that they have a company, a supplier that can actually supply them with a large quantity of this medicine if ever this will go to patients. They completed their QC and technical characterization successfully. They also demonstrated the effect in cells of rescuing the phenotypes, meaning in cells, this adenovirus gene therapy normalize the effect of NF2 cells in schwannomatosis. Also, the aim of this collaboration was to, first of all, create the gene therapy construct, but also testing it in non-human primate. As I said before, non-human primate is, let's say, the gold standard for gene therapy testing. Some testing can also be done in mice and rats, but the real deal is for non-human primates. And these, of course, are very complex and expensive studies. But they did the safety and biodistribution with no significant safety or tox. And the most important part that I bolded here is that they demonstrated very significant viral copy numbers in target tissue, meaning that the virus does what it's supposed to do, meaning distributing or regenerating the right copy of NF2. And this was observed in the vestibulocochlear nerve and also in the meninges. There is a ton of other tissue that still needs to be analyzed, but there is a very significant viral copy numbers. We don't know yet if that copy number level is meaningful from a therapeutic standpoint, but knowing that the virus does the job that it's supposed to do is very, very promising. They're also doing an NF2 mouse model. And this NF2 mouse model with the same AAV gene therapy, this time the mouse version, has also demonstrated the delivery of the vector to the NF2 tumors in that mouse. And also in the mouse, there is a demonstration of reduction in the number of Schwann cells that are in those tumors of those mice. So all in all, it looks very, very promising. And hopefully they will very soon complete the analysis report for all tissue. So there is tons of other analysis that are, because they need to see after the injection, where is the virus going? Is it going in the right tissue, which is the Schwann cells? Is accumulating in the liver? Is going to the brain? I mean, they need now to analyze every tissue that could be a target tissue for this virus. Also, to avoid side effects, for instance, of this virus going into tissue that were not supposed to be. The next step, as I said, is complete the analysis, but also if everything goes well and we have excellent results, the next step is to pitch the project to investors, create a larger group of investors, a large budget fund the next steps. And the next steps are the submission to the FDA. And in this case, it will be an investigational new drugs, IND, package that needs to be created. And it's very, very expensive to move to the FDA and then, of course, find its way to clinical trial. So very encouraging. Hopefully, more results will come later, and we'll know in the very first months of 2023. Another small update. This is something that CTF is not involved in, but Michael sent me those 3 bullet points that I want to share with you on their antibody drug conjugate program for meningioma. They scale up this candidate compound. They did the first rounds, and they also selected those top 4 candidates that are now, ready for, let's say, the next step of analysis and testing. So this is very, very early there. But the thing is that things are moving forward, and this technology of ADC is very, very promising in other cancer types. So hopefully it will be working also on meningiomas and NF2. There are no other programs that are looking at ADC. So it's a very good addition to the different approach that we're doing at NF2. I hope we'll keep you updated on this as well. Okay, an update on the platform trials on Intuit for NF2. As you know, Intuit for NF2 is a platform trial, so there is the opportunity here with one drug that enters the trial to basically monitor all the effects that this drug has on all type of tumors. So you see here the vestibular schwannoma, the non-vestibular schwannoma, the meningioma, and the ependymoma. Great thing is that once a patient is enrolled in the master trial, it goes in the natural history, and it's enrolled in one of this, arm of the trial and monitored with different drugs. So if there is response, it will stay on. If there is no response, it goes back and maybe can be, put on a second drug if the second drug is available. As you know, the first drug was brigatinib, and the brigatinib completed a first stage of 20 patients enrolled, and these 20 patients here gave a promising response rate. So there was a response rate of tumor tumor response, which was 28% for meningioma and 26% for non-vestibular schwannoma. It didn't really work on non-vestibular schwannomas. It didn't really work for vestibular schwannoma, and it didn't really work for ependymoma. But there is a promising, effect for meningioma and non-vestibular schwannoma. So now, because of these results, brigatinib is entering a second stage where there will be another 20 patients that will be enrolled in only meningioma and non-vestibular schwannomas to, let's say, strengthen the results. In general, though, the annualized tumor growth rates decreased for also vestibular schwannoma, non-vestibular schwannoma, and meningioma during the brigatinib treatment, and it was well tolerated. So these are all unpublished results that came from publications. Please keep confidential. Don't post it on Facebook, because these are very preliminary, but also very confidential for this group of as you are on the NF2 accelerator. But as I said, this is very, very, very promising. The second drug, so this is very also exciting. We have a second drug approved, and the second drug is called its name is neratinib, This is a drug that is being developed by Puma Biotechnology and has been approved both by Puma and Dana-Farber Institute. So the first patients has already been dosed at MGH, and other sites that are in the Intuit trials are already IRB approved. So they will start recruiting and dosing patients. Neratinib is a drug that from the mechanism of action will be looking at not hearing preservation, but tumor shrinkage. So again, is a multi tyrosine kinase inhibitor, meaning that these inhibitors target tumor growth and trying to stop the tumor or reducing the tumor size. So it will be monitored and patient will be randomized into, not randomized, but will be put on all type of tumors, and we'll see which tumor will benefit from this drug most. And hopefully we'll have a second stage like brigatinib, where we'll expand on what this drug is actually good for. So more to come in hopefully next year when we reach that 20 patients enrollment also on this second drug. Okay, last but not least, I want to put you on some other ongoing NF2 research. Actually, this is research that has been completed. I don't know if you remember, but Dr. Chun-Ling He from Georgetown University and Liam Laraba from University of Plymouth, they both worked on testing the hippoyaptase inhibitors. What they did is studying this pathway for NF2 that now is very well established, and they both tested as a tool compound, as a compound that targets this pathway, a compound from Vivace Therapeutics. So they both had an agreement with the company. We have talked to this company and we are in constant contact with them. Well, Vivace has started the clinical trials on NF2-driven mesothelioma in 2022. And they also told us that they are anyway interested in starting clinical trials for NF2 patients, so NF2 schwannoma or NF2 meningioma in 2023. So I would say the investments we made and the validation and the quality of these studies that were run at 2 different labs were very, very important to convince the company to start NF2 clinical trials in 2023. We're still in discussion if the trials will be in the Intuit trial or it will be a separate trial. So hopefully we'll get those discussions ongoing and we'll see the drugs approaching the clinic very, very soon. The good thing is that since this is already in mesothelioma, we'll probably have some benefit from an initial data on safety on that trial as well. Other things that I want to keep on your radar are other research that we are carry on. So we have an active grant with Jeremy Vite at UCLA on a very basic science project looking at micropinocytosis as a therapeutic delivery to cells. We have a very interesting collaboration with Tyron Porter, which is with the Focus Ultrasound Foundation. So it's co-funded by us and the Focus Ultrasound Foundation, looking at targeted drug delivery. So we're trying to shield the drug within a labile shield that can be break open at the site of the tumors by irradiating with this focus ultrasounds, and hopefully we'll avoid side effects at, for instance, other target organs. We have an ongoing grant with Isabel Logan at Oregon State on signaling pathways regulated by nitrated proteins. And very interesting, we had a grant with Philip Kulikov, who actually he was a postdoc in a Russian university in Moscow. But as you know, because of the war, he basically was able to also, with the help of the NF2 community, to be relocated in Germany, where he could actually, continue his research in looking at cytotoxic role of RAC1 to develop anti-tumor therapy for NF2. So this is thanks to the NF2 research community that we're looking for, helping Philip to move from Moscow, where everything got stuck, and move to Germany, where he could actually continue his research. So, and with that, I finished with my list of research and studies that we're doing for NF2, and we'll open for questions.
Salvo, we had two questions come up in the chat, so I'm going to read them. I'll give you one and then let you answer. Barbara's asking, if I recall, Elizabeth Castellanos Perez her work only works on those with mutation at exon 11 and not splicing variants. Is that correct?
It is, actually. And what they're doing in this new study, they're also looking at exon 8. So this is actually an expansion of that specific oligos that they did for exon 11. So yes, that's correct, but they're also looking at exon 8 in this new grant.
Salvo, Barbara also asks, are you seeing increased CPK numbers with brigatinib?
Not sure. Barb, what do you mean CPK?
That has to do with muscle and possible.
Oh, I don't. I probably have that information in Scott's deck, but I didn't capture it. So I wouldn't know. I mean, I don't want to say something that I'm not sure of.
Okay.
And Barbara, I put that note for Connie, and she asked me that if any of you brought up things that we couldn't address, to take note of it. So I took note of that for Connie.
Thank you.
You're very welcome. And Salvo, there's more questions. Nobody's raised their hand, so I'm just going to keep reading some questions. So can FUS be used on the NF2 tumor itself and not as a vehicle to deliver drugs?
Yes, actually, one of the let's say clinical application of focus ultrasounds is to ablate tumors, because they're very, very precise. Of course, the challenge with NF2 is the intertwined with the nerve, so it's not that easy to spare the nerve and use only for that. But that's definitely another application that can be used for. Sure.
Salvo, before we get off that, it's Barbara. If I could just add, I thought one of the concerns was you had to have a tumor pretty deep down for FUS versus right on the brain.
No, I think they can go they can reach vestibular schwannomas for sure. The problem is, if there is one problem that I understand, is that vestibular schwannomas are very close to bones. And irradiating close to a bone could actually cause a lot of heat, and that could be a side effect where instead of having soft tissue, hard tissue, it's a problem when you radiate. So especially when you need to cross past the bones. So that, I think, is one of the limitations of the technology.
Thank you.
Salvo, several more questions. You're getting lots of questions. Many thanks for the excellent presentation from Roger. His question is, there is more and more enthusiasm in the field using natural killer cells in treating solid tumors. Do you see any potential for NF2 tumors?
Yeah, yes. I mean, we haven't seen actually any ideas or proposal on using natural killer cells. Natural killer cells are very popular and very effective, I must say, on very aggressive tumors. Those are because the natural killer cells can spot malignant tumors right away. If you think about NF1 or NF2, there are benign tumors, and they kind of hide from the immune system, or let's say the surveillance that we have of this cell circulating. So they're not as a straightforward target for them. But I would love to see a proposal, because now they are becoming more and more sensitive. So these tools are probably the future.
Thanks. And Nissa said she apologizes if she missed this as you were speaking, but are any of the single kinase inhibitors shown to shrink NF2 tumors or just stop further growth?
It's hard to say. Normally, those single kinase inhibitors, they have multiple targets. So when we talk about a single agent, even the single agent can hit multiple kinases. It's very, very rare that you have a very clean inhibitor. And yeah, the idea is that it should stop and shrink, because what we see in tumors, that's the experience of this type of drugs in malignant tumors, is a lot of shrinkage of these tumors. But NF2 is always different. So we've seen some kind of shrinkage now with brigatinib, for instance, which is a kinase inhibitor, and other, but it's limited, and we don't know how long that shrinkage will last. Like in the case of selumetinib, for instance, for NF1 patients, that's another kinase inhibitor, and the kinase kind of hit a plateau, so they reach an effect, and sometimes it's very difficult that you eradicate the whole tumor. It may stop at some kind of degree of reduction and plateau there, but it's really, really difficult to predict, and it's very variable patients to patients.
Lots more questions, Salvo, so I'll try to go. Natalie said this is very exciting. So the neurofibromatosis trial is looking at multiple tumor types to be treated. Do they have a feeling if it will work on any particular tumor type yet, or just all our targets?
Well, Natalie, this is exactly why we have the Intuit trial, that now with platform trials, we are looking at all tumor types. So the neurofibromatosis drug is going to be tested at least 3 patients per tumor type, and we'll, and the protocol foresee that you recruit 20 patients. So there will be probably, maybe more patients on vestibular schwannoma and meningiomas than ependymoma for sure, but at least we'll see also the ependymoma response. And by the way, every patient is primary objective is the primary tumor, but they also be monitored on the other tumors, because if there is an effect on a secondary tumor, they will move and be put on that secondary tumor, let's say arm. So the beauty of the let's say the platform trials is that we'll monitor all of them, and then the drug will be work, whatever tumor type works, the drug will be put on.
Salvo, Dongjun has several questions, said, great talk. Thank you very much. So a couple of fundamental questions. One, how sure the AAV mediated gene delivery system is specific to Schwann cell population?
Yeah, great question. So the AAV that was developed for NF in the context of NF1 is very specific. It's actually more specific than anything has been published so far. This is still unpublished, and I think it will come up very, very soon in publication. But they achieved a level of specificity for Schwann cells that is very, very high, higher than anything that has been published. The other very good peculiarity of this vector is that it doesn't accumulate into the liver. So normally all of these virus and capsid, they go where they need to go with more or less efficiency, but then they get delivered, they accumulate in the liver. What we're looking at is that this virus will not accumulate in the liver, so there will be more in circulation. We still need to know where it goes and how it gets excreted. So probably, via other meanings. But this is the good news so far, is that we know it's very efficient for Schwann cells and it doesn't accumulate a lot on the liver.
Salvo, I don't know if we'll get through them, but I'm going to try how to overcome the CRISPR-Cas off-target effect is another question.
Yeah, that's something that we need to they need to test. So what I want to explain is that they tested the carrier, but we the carrier was tested in cells with the payload, and that actually worked. So in cells, the concept really works. Now the question is it working also in mice, for instance, or and do we see the same delivery in non-human primates? Those are the two next steps that are happening. In concomitance, we're also using a different payload, this case the NF2. We are funding a larger application that is looking at efficacy of the virus, the payload in cells, in mouse, and hopefully will also go to the non-human primate. The proposal also has a part where they're looking at new viruses. So even if this is the best so far, they have also a proposal to screen another 40 different AAV.
I'm still several. Salvo, we'll see what we can get through. Reduction in the number of Schwann cells in DRG of treated mice. What if the AAV NF2 can damage the other functional peripheral nerve myelinating Schwann cells?
Yeah, and we'll learn about those because they're looking at so many. I think there is at least 50 different tissue types that are looking at. So the analysis is not complete. What I reported about the DRG is because that was the target that the AAV was supposed to target. So that actually works. Now we need to see where else this virus has gone and in what quantity. So this is the very first things that you look at. Is the virus doing what it's supposed to do? Yes. And what else? And that what else is big question mark, but we'll have full details because there is a very long list of tissue types that is going to be analyzed.
Okay, I think we have 2 more questions in 2 minutes, so this is perfect. Natalie's asking also, when do they anticipate human trials taking place for NF2 gene therapy? Is this a type of CRISPR therapy or another type of gene therapy?
Yeah, no, I want to make clear that we are very, very far still from the clinical trials. These are concepts, and before we get to clinical trials, we're still few years into the making. This, for instance, is let's say more advanced for NF1 because we already are planning the non-human primate testing than it is in NF2, where we are starting now, even though we are taking advantage of that study, but we're still far. This is nothing that this is not a study that is going like in the next 2 years, 3 years in clinical trial. There's a lot of things happening before that. But it's hopeful, and we are on the right track. So that's the positive note.
Tracy is asking, Tracy Galloway, is there any way we can encourage the FUS Foundation to please try their technology on peripheral tumors?
Yeah, actually, they're very interested, Tracy, to do that. And so maybe it's a second collaboration that we can have with them. They're very, very eager to test this technology on potential new therapeutic areas. So absolutely.
I'm going to be respectful of time now that we're at the top of the hour. I think we missed a couple of questions, but Connie's going to get the transcript. So if we miss some questions, we'll try to get those answered for you guys. But because it is now 2 a.m. Salvo's time, and it's getting late for all, I think I'll get ready to wrap up. Salvo, you're getting a lot of thank yous in the chat. So thank you for this. I will also just want to thank all of you for joining us, making time to be here. I hope you got a lot out of this. I always love hearing how many brilliant people are out there working so hard and passionately for our NF community. If you indicated any interest in becoming an NF2 accelerator or learning more about the program, Connie will get in touch with you soon. And so that will happen. And then Tracy, you're saying I can keep going a little bit since this is my first time. Should I?
No, I'm saying we keep going with our research and our push.
Oh, got
ahead.
you.
Go
Okay,
ahead.
sorry. I was like, should I keep going? I'm real strict on time with things. I apologize. Okay, so if you didn't indicate an interest in the program, Connie will get in touch with you. And then I just want to also remind you all that we'll have a brand new calendar of our monthly meetup scheduled for 2023 very soon. So Connie will get that out to you all. And we just, from all of us here at CTF, we just want to wish you a warm and wonderful holiday season. So thank you again all for being here, and thank you, Salvo, to you as well. So have a good evening, everyone.
Thank
Take
you,
care
everyone.
of yourselves.
Yes.
Thank you.
Thanks
great
all.
evening.
Bye.
Bye-bye.